name is Lorina Naci and I'm Associate Professor of Psychology at Trinity College Dublin. So I'm here from Ireland today as one of the investigators involved in this amazing study, and Brian Lawler, also from Dublin, is here with me. So a little bit of background as to why I'm here today. Around 2018, Trinity College Dublin became the fifth site of the PREVENT study in Dublin. So we joined about maybe 4 years later than when the main study was founded in the United Kingdom. And I had heard about how dedicated and incredible the group of participants in the PREVENT study were, and we've heard these anecdotes of people just participating, being extremely engaged, and just being such a pleasure to work with. And then when we put an advertisement to run the study in Dublin, we were looking to get 100 participants to recruit into our site to then pull together with the other sites of the study to create the ultimate number of 700. And we were a little bit intimidated by the success of our colleagues in the United Kingdom in recruitment. And then we got 600 people coming forward to take part in the study for 100 places. And so the first challenge in the study was actually just going through the list of inquiries and interest, and finding a way to, in a fair manner, to select the 100 candidates. So we ran a number of participant events in Dublin. I don't know if anybody from our participants is here today, but on behalf of the Dublin team and our participants as well, I want to thank you for being such a tremendous group to work with. And that enthusiasm of people participating in the study was one of the reasons why I fell in love with the study when I first joined the PREVENT team. So thank you very much for really helping us out and truly joining forces to try and understand the earlier signs of Alzheimer's and dementia, as it manifests in the brain and body. So today I will touch on some highlights on work on cognition and lifestyle, also including some findings, particularly on the cognitive side, from Professor Karen Ricci, who is at the Inserm Institute in France, who couldn't be here today. So as you've heard, we now understand that we need to take a lifespan approach to understanding of dementia, and that the earliest signs or the earliest starts of dementia neuropathology probably start decades before clinical symptoms manifest, and we think that midlife is a really appropriate time frame for starting to investigate the objective signs of disease. And Craig will probably tell you that when he first said this sounded very disruptive to the funding community and almost everybody, in 2014 or even earlier. But now this is relatively well accepted, and in large part through the work of the PREVENT study. And so, as you are now very well aware, the whole point of starting this early is that by detecting the earliest signs of disease before the clinical symptoms appear, we have the highest chance of understanding who is at greatest risk and helping to deliver new treatments that may even slow or prevent the disease in the future. So this is tremendously important. Also, since the time when the PREVENT study started, there is a greater acceptance in the scientific community that indeed there needs to be a lifespan approach, and a number of other risk factors have been suggested through big aggregation of studies or meta-analysis, not through that kind of really in-depth, rigorous scientific approach that we're taking here. So basically now we understand, there's a kind of understanding and agreement in the scientific community that there needs to be a lifespan approach, and that risk factors start to impact cognition and the brain very, very early, probably as early as from birth, when we have certain genes, including ApoE, that is impacting the way that our brains develop and are susceptible to risk. And then a number of risk factors and have been suggested for midlife in particular, and then later life. And that the writing is probably quite small, but those include hearing loss, obesity, hypertension, traumatic brain injury, etc. in midlife. And they are risk factors that are currently being investigated, and they're not the ones that we'll look at here. But it just goes to speak to this idea that there are certain lifestyle choices that can present risk, including traumatic brain injury, where that is avoidable. But additionally, the multi-domain aspect of this is that there is also a number of protective lifestyle factors that we have a choice about, including education in early life. And then what we've known from studies in later life include physical activity and cognitive and social activity. And what we're doing in PREVENT now in some of our recent studies is trying to bring this multi-domain approach into trying to understand the interplay between risk and protective factors. And particularly in the period of midlife there, we don't really know, while we know there are some risk factors, we don't know how those protective factors might offset the effect of risk in that early time period. So at a very highlighty sort of level, I'll just touch on some of the findings on the impact of risk factors on cognition in midlife and the impact of protective lifestyle factors on cognition in midlife and how that may offset risk factors. So again, as you've heard today in John O'Brien's talk, we are looking very, very early in a time period where clearly there are no memory problems, there are no clinical manifestations of the symptoms of dementia. So individuals are healthy, and we're looking for objective indicators that really it is the case that cognition as one of the manifestations of brain health is objectively changing in this group. And similarly looking at whether we can find objective measures, objective indicators of the effect of protective lifestyle factors at the group level. And so the approach that we take is that we measure cognition, that I'll tell you more about in a minute. We particularly focus on this very well validated risk factors that have a really dramatic impact in later life. So we know they are very important to look at their manifestation in early life, the ApoE gene, family history of dementia, and the cardiovascular incidence of dementia aggregate score, which includes genetics and biological sex, but also includes a number of cardiovascular-based risk factors. So a lot of people think of it as predominantly modifiable. So let's look a little bit at the impact of risk factors on cognition in midlife. So what do we mean by cognition? Let's just to break down a little bit the jargon that we use as scientists every day. When we think about cognition, we think about the process of acquiring knowledge and understanding. And there are many domains of cognition that can be investigated, and where indeed in later life we see big effects of dementia and Alzheimer's, including attention, memory and learning, abstract thought, reasoning, computation, decision-making, problem-solving, spatial analysis, comprehension, and production of language. And with cognitive decline, what we are trying to understand with cognitive decline is really what is the central clinical concern in relation to dementia, and that is considered necessary for diagnosis. And when we're looking at cognition in midlife, we're not necessarily looking at decline, but we're looking at whether individuals who have family history or who have the ApoE allele 4 gene are doing overall worse than individuals who don't have those risk factors. And then the cognitive tests that have been used for diagnosis to date, prior to the work of PREVENT, have been derived from studies of persons with advanced brain changes, so in later life. And therefore, one of the challenges of the PREVENT study when it started was the detection of potentially silent cognitive changes in younger asymptomatic persons who were at risk And this has required a development of alternative methods, and this is the work of Professor Karen Ricci, who couldn't be here today. And she has been very instrumental at devising the kinds of tests that you have all taken, particularly through the Cognito battery that is trying to assess changes in different domains of cognition that are subtle and that may not be the same ones as those that we see in individuals who are presenting in the clinic with memory problems that are related to mild cognitive impairment or various manifestations of dementia. And in particular, the use of specialized spatial memory and spatial orientation tests has been highlighted and forefronted in PREVENT because it targets areas of the brain that are known to first manifest the Alzheimer's disease neuropathology. And we use several of these tests in the PREVENT cognitive battery, including visual binding tests and the four-mountain test and visual reality. And there are people in the room that actually are still recruiting or experts in these tests that will tell you more about them if you want to discuss further. But I suppose the main point to highlight here is the sort of challenge and the reverse engineering that we have to do when we have to try and detect the earliest signs of a hidden neuropathology. And so I'm taking you through the thinking process here that was employed and that has been a quite fruitful one. So on the screen is our slices or renderings of the brain in from the left to the right, the earliest to later documented stages of the Alzheimer's disease type neuropathology from postmortem brains, from neuropathological findings. So here we're not looking at the same individuals over time, we're looking at individuals who may have been different cohorts at earlier age groups, maybe 50 or 60 or 70, and with older age and more advanced pathology, we can see more widespread changes on the picture here to the right. That's all showing the warm-colored areas. But what we know from these neuropathological studies is that the transentorhinal regions of the brain, so the medial temporal lobes that are implicated, they're very important for memory as well as a number of other functions, including spatial cognition, show the earliest signs of the accumulation of aberrant proteins that are hallmarks of the neuropathology of Alzheimer's disease. So Karen Ricci and colleagues, when they're trying to detect then these hidden changes in the brains of healthy individuals, thought that perhaps looking at the kinds of functions that these brain regions underpin is a good way to go to find where those changes might be occurring in individuals who otherwise are healthy. And so I will just highlight some of the findings from this work on cognition to date. So what the PREVENT study overall has found is that participants with a parent who has dementia have poor visual memory the closer they are to the age at which the parent was diagnosed. And this one was one of the earliest findings. It was surprising because it wasn't necessarily known from the literature that the visual aspects of memory would be implicated in early changes, but it makes sense when you think of the kinds of regions that are showing neuropathology the earliest accumulation of aberrant proteins. Participants with a high dementia score, the CATE score, have greater difficulty in recalling the names associated with faces. So this is a form of relational memory. Participants with a parent with dementia have increased difficulty recalling spatial organization of objects, so recall of a description of an object, especially if they have a lower hippocampal volume to begin with. So again, we're picking up a theme of spatial memory and spatial cognition being some of the earliest changes, imperceptible at the individual level, but nevertheless very significant at the group level. and that persons with a high risk of dementia have difficulty with tasks involving spatial analysis and spatial memory. And some of these results have been picked up through more specialized tests that involve, one of which is called for mountains, which most of you in the room will have participated in, which is looking at 3D images where the challenge is to detect changes in the scenery that rely on spatial orientation and spatial memory. And what we've seen is that patients with a high dementia risk have greater difficulty with this test. And taking this one step further, in Coco's work, who is here in the audience today, the visual reality test, where we're looking at a more realistic assessment of spatial cognition and, so to speak, testing our GPS system of the brain in real life, we also see poor performance in people with a family history of dementia and genetic risk. So the take-home message from these studies is the importance of really thinking outside the box and looking outside of the domains that are implicated in late life when it comes to identifying early changes that might be kind of the sentinels and the earliest indicators of the disease. And so the next point that I want to touch briefly upon is then, is there anything that we can do to stem this type of changes when we're thinking about protective lifestyle factors? And what are we even talking about when we're talking about protective factors? So with protective factors that are related to lifestyle choices, we're thinking about activities, choices that we make that we can modify that are thought to imbue cognitive reserve or the capacity of the brain to withstand the effects of insult or injury or neuropathology, and to keep performance and to maintain cognition even though there might be underlying neuropathology. So cognitive reserve acts as a buffer to a potential incipient neuropathology. And some well-established factors in reserve, cognitive reserve contributors, as they're known, particularly in the work in late life, are education. So particularly the way we measure education is education in early life, although recent studies are shown that even adult education, especially in populations that have been traditionally underprivileged and have had lower amounts of education in their life, can really make a difference. With stimulating lifestyle activities, we're looking at cognitive, intellectual, and physically stimulating activities, and measuring a number of domains, including socializing with friends, playing a musical instrument, learning a foreign language, practicing an artistic pastime. Musical instrument is there twice, sorry. Engaging in physical activity and traveling. So these are avocational activities that we do in our free time, but it turns out that also the effort that we put in our vocation can make a big difference in terms of cognitive reserve, and we measure this through occupational attainment. This includes both the complexity of the job and the nature of the job itself, but also complexity that comes with managerial responsibility. So being in charge of people is also adding to that index of occupational attainment as it pertains to cognitive reserve. So I'll tell you a little bit about some of our findings. We measure this reserve contributors in a very rigorous way through well-validated instruments, and one that is internationally validated and has proved very productive is this instrument called Lifetime of Experiences Questionnaire, which will depict the cognitive reserve contributors in different stages of life. And here we have focused in midlife because we're particularly interested in people in this age group that we're studying and what can actually be changed today as we are continuing to make choices. So we think obviously what happened in young adulthood is really important, but are there modifiable choices that we can make today? And so the first thing that we have found is that the years of education, the higher the amount of education is strongly associated with stronger cognition in the PREVENT cohort. This is not surprising. It has been found in other studies as well, and it is not novel, but it is yet another indication of the importance of education in midlife. And because education is predominantly taken, kind of counted for our activities in early life, we try and account for this and control for this when we're looking at the other contributions, just so to make sure that, you know, it has been set aside, the contribution of education. So if we leave aside the education, what else can we see then? We see that all of those socially, physically, and intellectually engaging activities that I mentioned are also very protective in terms of their positive, strong, significant positive association between the amount of engagement. So the more engagement in these activities, the higher and the stronger the memory performance at the group level of our participants, so that is you and everybody else involved in the PREVENT cohort. We also see that this particular contribution of stimulating lifestyle activities to cognition is particularly important for people who have a risk factor for Alzheimer's disease. And in this group, that one of their risk factors is a family history of dementia. So the red line here in the last graph is showing the strong positive association, particularly for the group who has a family history of dementia. So they seem to benefit from higher engagement in this engaging activities in relation to their cognition. And we think this is that perhaps, obviously the PREVENT study wasn't initially designed to look at the protective effect. So we have to do more work to follow these results up. But one of the potential underlying mechanisms at work could be the fact that individuals who have a family member of dementia, many of them are involved in caregiving duties, and we know the effects of caregiver burden on brain health, and part of that comes through an environmental deprivation, loss of opportunity in terms of being engaged in stimulating activities, having the opportunity to engage in things other than caregiving, or at least having reduced amount for those. And so one of the potential mechanisms is that the higher engagement in these activities is counteracting that potential negative effect of environmental deprivation in individuals with family history of dementia. And that's why we find this particular reserve contributor to contribute to cognition in individuals with family history. And so another really interesting area that is growing of importance now, 10 years later from when PREVENT was initially designed, and that is going to lead to further studies in PREVENT. And in fact, one of our investigators, Graciela Munoz-Terres, is following this up with more work, is looking at biological sex-specific differences in dementia. And the data is incredibly similar and robust across the world. But here, for example, this data from the United Kingdom, where it's quite similar to the United States, Ireland, etc., as well as LMIC countries and the rest of the world. Women seem to be at grist risk twice as higher. 2/3 of Alzheimer's and dementia cases are female. And this is not only explained by age, although age with women living longer is potentially contributing to this effect, but as shown in the second graph here, provided by the Alzheimer's Association of the United States, in two groups, one with mean age of 45 and another one with 65, we still see a very similar proportion of females and males with Alzheimer's disease. So the underlying mechanisms are a big research area at the moment, including hormones, including genetics, and various factors that build a very complex biological mechanism underpinning this disparity in prevalence in females versus males. But also notably, we mustn't forget that around 2/3 of of carers for individuals with dementia are female. 1/3, around more than 1/3, are daughters. So in addition to being at risk increased risk through a biological mechanism, females are also exposed to higher caregiver burdens, so they're suffering through another form of risk factor on brain health, which potentially is interacting with the biological mechanism. So there's a complex picture building around potentially why females are at higher risk for Alzheimer's disease. And we don't have the answers yet, but hopefully more of this in the future work in dementia. But one thing that we see in our data with regards to reserve contributors is that when we separate females and males, we see a disproportionate effect of occupational attainment in males versus females. Here we see in salmon color are females. I'm not being very good at managing the pointer, and males are in blue. And so we see that for females, higher occupational attainment is associated with stronger cognition in episodic and relational memory. We don't see this to be the case for males. So this is really interesting and suggests that this higher occupational attainment is better for females in midlife. And when we also look at particular females who have also the genetic risk factor for Alzheimer's disease, we see this effect even more pronounced, that APOE carrier females are even more, have a stronger relationship between higher occupational attainment being associated with higher cognition in episodic and relational memory, and this does not seem to be the case for non-carriers. So this is really intriguing to us, and we are following it up. One of the areas that we want to follow up is, of course, the effect of the menopausal transition in this group, because obviously the loss of the estrogen and other hormonal changes in the period that's covered in the PREVENT, the age group, will necessarily lead to loss of protection in the female brain, and how is that driving these results or potentially affecting the brain health changes. So again, this study wasn't designed for this. I can only offer some tentative suggestions, because we do need to get more data and look specifically into this particular question. But suggestingly, when we look at the postmenopausal females that are APOE carriers in this group relative to the ones that are non-menopausal, we see, so that's the purple line here, we see that this is the group. So I call this group the triple risk threat group. The females, their APOE, and their postmenopausal. And this is the group, again, that seems to be most benefiting, or at least strongest association between occupational attainment and stronger cognition. And so in terms of the wider implication, as we follow this up, I think this is really important, also because it could be a potential mechanism for protection and cognitive reserve in a group that, especially around midlife, is very vulnerable to loss of occupation or moving away from the workplace for various reasons. So this potentially could suggest a policy change to really try and retain females in the workplace in this age group in order to protect the brain health of females, especially genetic risk and postmenopausal as a vulnerable population. So I just want to conclude this very brief highlighting of some of the findings in these domains by saying that we do see objective evidence that risk factors do impact cognition midlife at the group level. So these are not individually relevant level for each and every individual that is involved in PREVENT, but at the group level. So we're seeing that the risk is already starting to impact the brain. But the good news is that there are things that we can do about these changes, and that protective lifestyle factors really can help to build cognitive reserve in midlife, and that the reserve contributors are several. So there are several ways and mechanism pathways through which we can help to build cognitive reserve, including educational, vocational, and avocational activities. and that more frequent engagement with these protective factors lead to better cognition, especially in those with family history of Alzheimer's disease, and that higher occupational attainment is associated with stronger cognition in females, especially those that have the genetic risk through ApoE allele 4 carriership. So thank you very much for your attention. It's a pleasure to be with you today.